Today we're going to be looking at the nitrogen cycle. The nitrogen cycle is a very important topic because nitrogen itself is composed of about 78% of air. So now I'm going to tell you the processes that remove this nitrogen from the atmosphere, and they occur in many ways. So the first one is nitrogen fixing microorganism. So this is when free living bacteria like the Azobacter, the Anabaena, or symbiotic bacteria like the Rhizobium, which is usually found in the root nodules of leguminous plants. So this converts atmospheric nitrogen into soil nitrogenous compound. Yes. So the action of thunderstorm is the second one. So you know the first one is nitrogen fixing organism. So the second one is thunderstorm, the action of thunderstorm. So this process remove nitrogen from the atmosphere. Yes. So the first one is nitrogen fixing organism, right? This, and I said, is when a free living bacteria or symbiotic bacteria convert atmospheric nitrogen into soil nitrogenous compound. Yes. So the second one is the action of thunderstorm. So during thunderstorm, nitrogen in the atmosphere reacts with oxygen to form nitric oxide. So during thunderstorm, nitrogen in the atmosphere reacts with oxygen to form a nitric oxide, which then dissolve in the rainwater, listen, to form nitric acid. It dissolves in rainwater to form a nitric acid, and this acid then enters the soil. So after it enters the soil, it combines with the mineral salt in the soil to form a nitrates, Yes, so now I'm going to give you the process which converts soil nitrogen to atmospheric nitrogen. You know, the first one I gave you is the process which remove nitrogen from the atmosphere, which is nitrogen fixing organism and the action of thunderstorm. So this one, I'm going to give you the processes which converts soil nitrogen to atmospheric nitrogen. So this is done by a process known as the denitrification through bacteria called the denitrifying bacteria. So the denitrifying bacteria did this process through the process of denitrification. An example of this denitrifying bacteria is the Pseudomonas denitrificans. Yes. Note, denitrification occurs in the presence of low oxygen or no oxygen at all. Yes. So this denitrification occurs in the presence of low or no oxygen at all. And not the following definition of these terms I'm going to give you. I'm going to give you the following definition of these terms I want to use. It's part of the nitrogen cycle as a whole. This topic is very short. So the first one is putrefaction. Putrefaction is also known as the decay. Listen carefully. The decay that you guys hear all about, decay. So decay is also called the putrefaction. It's the process whereby nitrogen present in living organism is converted into soil nitrogen or re-enters the nitrogen cycle. So listen, it is the process whereby nitrogen present in the living organism is converted into soil nitrogen. So the nitrogen present in we humans, like we living thing, is converted into soil nitrogen. So that's what brings about decay after death. Yes. So let's look at the ammonification. So the ammonification is the process by which proteins are broken down into ammonia. Yes, that's the simple definition. There is nothing there. So let's look at the nitrification. Listen, nitrification is the process whereby ammonia and other nitrogenous compounds are converted to nitrites. Nitrites, remember, not nitrates. So this is the process whereby ammonia and other nitrogenous compounds are converted to nitrates in the soil. And it is also defined as the process whereby nitrates is converted to nitrates. You see there, nitrates and nitrates are different. So it's also defined as the process whereby nitrates are converted into nitrates. And it occurs in the presence of high amount of oxygen. You know, I said denitrification occur in low or no oxygen at all, but nitrification occur in presence of high amount of oxygen. Yes. So note, denitrifying bacteria that converts ammonia to nitrates is called the nitrosomonas, while the one that converts the nitrates to nitrates is called the nitrobacter. Yes. So denitrifying bacteria that converts ammonia to nitrates is called the nitrosomonas, and the one that now converts the nitrates to nitrates is called the nitrobacter. Yes. So that's just it though. This thing is not really hard. So let's move on to the next one. Before we move on to the next one, note that the denitrifying bacteria are anaerobic bacteria and the nitrifying bacteria are aerobic bacteria. You know, I said denitrifying bacteria occur in the presence of low or no oxygen. So they don't need oxygen, right? So that makes them anaerobic. And the nitrifying bacteria occur in the presence of oxygen. So that makes them the aerobic bacteria. So now I'm going to tell you the meaning of water culture. There's what we call the water culture, which is also known as the hydroponics. So this water culture is the method of growing plants using distilled water. Look at it, the method of growing plants using distilled water containing necessary elements in a correct portion needed for the growth of plants. Very simple. Containing the necessary elements in correct portion needed for the growth of plants. So, water culture was discovered by a scientist called the Knox and Sachs. So that's the scientists that discovered the water culture. Yes. So let's look at the precautions to carry when carrying out this culture experiment. So this is the last thing that we're going to talk about for this class. So the precautions to take when carrying out this culture experiment. So the first one is all culture vessels must be painted black or covered with a black paper. Yes. So this is to prevent the growth of algae. Yes, so the second step is that all culture vessels must be sterilized to avoid contamination. Good. So you must sterilize your culture vessels to avoid contamination. Yes. So the third step is that healthy growth seedlings of the same size and age must be used. So you cannot use a bad one. Always select the healthy one with the same size and age. Yes. Then let's talk about the fourth one, the fourth process. Dry cotton wool or a stopper should be used to hold the stem to prevent the rotting of the stem. A dry cotton wool or stopper should be used to hold the stem in place to prevent rotting of the stem. So the last one is that air or oxygen must be blown in daily in the culture solution through the aerated tube. There is what we call the aerated tube. It's in the culture experiment. So air must be blown into it daily to replenish the oxygen used by that plant, to keep that oxygen intact in the plants. So that is all that we're going to talk about in this class, and the topic is the nitrogen cycle. So I just explained it briefly. So do not forget to like, subscribe, comment, okay? So see you guys in my next class.